Turn on the light.
Christine, are you going to cue me?
Say that again.
Are you going to give me a cue?
You can start anytime. I would say wait till 11, but yeah, I'll send you a message.
All right. Great.
Camera, thank you for the invitation. I really appreciate it. I hope everything is fine on your side.
Absolutely, Danielle. And I'm very much looking forward to your talk. I see this as a little bit of a new direction for your work as well. So I'm very interested in seeing where you're going to take us today. I think it's time for us to begin. So I'm going to introduce you, if that's all right. Good morning. Welcome to today's Cannabis and Hemp Research Forum hosted by the UC Davis Cannabis and Hemp Research Center. I am co-director Cameron Carter. The purpose of this monthly forum is to inform our campus community, members of the cannabis and hemp industry, and the general public on cannabis and hemp research being done at UC Davis, and also provide an opportunity for external guest researchers to share their expertise. As today's presentation is underway, you'll be able to pose written questions using the Zoom Q&A feature. Our presenter will save time at the end to address your questions. This presentation is being recorded, and the recording will be made available in the next few days on the Cannabis and Hemp Research Center website at cannabis.ucdavis.edu under the Education tab. So we're very fortunate today to have as our speaker Professor Danielle Piomelli from UC Irvine. Dr. Piomelli studied pharmacology and neuroscience with Schwartz and Kandel at Columbia University and Paul Greengard at Rockefeller University. He joined, after a period in San Diego, he joined the faculty at UC Irvine, where he's now the Louise Turner Arnold Chair in Neurosciences and Distinguished Professor of Anatomy and Neurobiology, Pharmacology, and Biological Chemistry. He's the author of over 400 peer-reviewed articles. His research is focused on understanding how endocannabinoid substances and other lipid-derived signaling molecules affect the function of the brain and other organs. And more recently, he has focused on the impact of cannabinoids on adolescent brain development. The title of today's talk is Long-Term Consequences of Adolescent Cannabis Exposure. It's time to take a fresh look. Thank you, Dr. Piomelli.
Thank you very much, Dr. Carter, for the nice introduction. Yes, I will be talking about cannabis in adolescents, particularly about work that we have been doing in the last 3 to 4 years in this area, trying to understand how adolescent cannabis use can impact the endogenous cannabinoid system, and by doing that, impact brain health and function. But this is an overview, quick overview of my talk. I am cognizant of the fact that not everyone necessarily is an expert in cannabis and other things. I will be giving a short introduction about cannabis itself and its chemical constituents. For many, this will be repetitious, but I hope you won't mind if I do so for those who are less expert in this field. I will also introduce the endocannabinoid system. And finally, I will share with you new data on the lasting consequences of adolescent cannabis use, which I believe should make us rethink the way we are approaching this issue. So let me start off and to go, for those of you, again, who are not experts in this, cannabis, of course, here is the plant, the inflorescence and the leaves of the female plant, from which folks over the centuries and actually millennia have been producing drugs, substances such as marijuana, shown here, or hashish, shown here. Now, the plant itself comprises a large number of, produces a large number of chemicals, the structures of which, of a few of them are shown here. Over 140 cannabinoids, phytocannabinoids have been identified, and these compounds share this terpenophenolic nucleus that you see here, for example, in the case of tetrahydrocannabinol or THC. In fact, cannabis, excuse the pun, is actually a chemical plant in the sense it's really a chemical industry, and the manufacturing aspect of the part of the plant is the glandular trichomes, which contain enzymes of a biosynthetic pathway that resembles that gives rise to fatty acids in mammals, although is different, and is complex, as you can see, a complex pathway that leads to the production of two phenolic acids, delta-9 tetrahydrocannabinolic acid and cannabidiolic acid, along with others. So from a legal perspective, these two terms are often used, marijuana and hemp. And for those of you who are confused about this, which is quite possible because it is a fairly confusing nomenclature, marijuana and hemp both refer to the cannabis plant. It's the same plant, but to the federal government, marijuana is anything that has more than 0.3% THC, and hemp is anything that has less than 0.3% THC, and presumably also high CBD. The two are mutually exclusive. The synthesis goes in one direction or the other. So how do we get from THCA, which is what the plant produces, to THC? We get there through loss of this carboxylic group. This decarboxylation is catalyzed by a variety of agents. Most commonly used is heat, of course. And for example, this is how from the cannabis plant, you get to hashish, you heat it up and you produce this extract. And the product, delta-9 tetrahydrocannabinol, you can see it here, this benzoypyrene molecule with this fairly long side chain, alkylic side chain. And this is a fairly unstable molecule to a certain extent. It is eventually converted over time, over months and years, into cannabinol, which is actually, you can see cannabinol, of course, here has the aromatic ring here. And this is actually useful from an archaeological perspective. And for those of you who are interested in this, for example, this is a tomb that was excavated in the city of Turpan in the People's Republic of China a few years ago. And this basket was discovered containing about half a kilogram of cannabis. And they could have been hemp, right, with not so much THC, or it could have been marijuana, according to the federal government, with a lot of THC in it. And what the investigators did to make sure that was actually biologically active, physiologically, psychologically active, they used mass spec to determine the presence of cannabinol. And this was also done not so long ago on an altar discovered in a temple in Israel, in the city of Tel Arad. And yes, these ancient Israelites used to burn cannabis in their temples. It's an interesting thing some of you may not know. Anyway, moving on, the body actually does not transform THC, carboxylic acid, into THC, cannot do that, but it does transform THC into, first into an hydroxylated metabolite through the cyclone P450 system in the liver and other organ systems. And this hydroxylated compound is further oxidized into an aldehyde and then eventually into a carboxylic acid, which is 11-nor-9-carboxy THC. There is a mistake. This is 9-carboxy, not 9-carboxy OH THC. Sorry about that. This is important because actually the pharmacology of cannabis, not just the pharmacology of THC or CBD, is also pharmacology of their metabolites. And I think 11-hydroxy-THC, which is equally potent and possibly more efficacious than THC is actually produced differentially in a sex-dependent and age-dependent way. So we know, for example, in females of mammals, mammalian females, so in women as well as in female rats and female mice, the transformation of THC into 11-hydroxy-THC is much more efficient. There is much more, and that is produced. These are levels of THC, hydroxy, 11-hydroxy-THC in circulation in women. They work out of the lab of Ryan Vendry, and this is in men. Actually twice as much 11-hydroxy-THC is produced by women than by men. And because the pharmacology is different, they could at least in part explain the interesting sexual dimorphisms that we observe, that are observed in the effects of THC. In fact, adolescent animals, we don't know about adolescent humans, but adolescent animals seem to behave like women do, in that they seem to have much greater production of 11-hydroxy-THC than adult animals, adult rodents do. So that is what I wanted to share with you about cannabis. It's a chemical constituent, as a reminder. Then now I'd like to go into the endocannabinoid system. So what is the endocannabinoid signaling system? In a nutshell, is first of all the port of entry for THC into the body. This is how THC interacts with the human body, the body of any other mammal, is through binding to and activating cannabinoid receptors, which are part of the endocannabinoid signaling system. There are two cannabinoid receptors, CB1 and CB2. I will mostly talk about CB1, mention only CB2. And these receptors are activated by a set of endogenous molecules, which are called endocannabinoids, and they are lipid derived. So they derive from precursor in membranes through a series of enzymatic reactions that are started upon demand. And the two main endocannabinoids are anandamide and 2-AG, and I will triacetyl glycerol 2-AG, which I will show you in a few minutes. To make these endocannabinoids and to destroy them, you need proteins, enzymes, transporters, and these are the protein components of the endocannabinoid system. And I will also describe them to a certain extent. So CB1 cannabinoid receptors, in which I think we should keep us focused, because those are the receptors that most likely are involved also in the adolescent effects that I will describe to you later on. They were discovered in 1988 by Lynn Howlett and Bill Devane, and then cloned 2 years later by Tom Bonner and Lisa Matsuda. And what you're seeing here is actually not a model. This is the actual structure determined by X-ray crystallography of the receptors, which was published a few years ago. It is a very prototypical G protein coupled receptors in many ways, with one important exception that I'd like to point out, is that the binding site for the ligands is not on the external surface of the receptor. This would be the external surface, and this would be the intracellular surface, the membrane being here, but rather is located inside the membrane. And that is something that fits very well with the biology of the pharmacology of THC and the biology of the endocannabinoids. In fact, work by Alex Makrianis several years ago showed that not so much THC, but a similar compound called CP55940 is able to bind to cannabinoid receptor by lateral diffusion. So it enters the membrane, as you would expect from a hydrophobic molecule, and it laterally diffuses and binds to the receptor in this particular way. And this is how also, I personally believe, endocannabinoids bind to the receptor. So the receptor per se, as I said, is not especially different from others. So an agonist can activate G proteins. These are these trimeric transducing proteins, alpha, beta, and gamma subunits. Upon binding, the beta gamma subunit have all sort of effects on ion channels. And the two main effects that have been characterized are the closing of calcium channels, which you see here, and the opening of potassium channels. In neurons, this, of course, would result in a decreased neurosecretion, less calcium, less neurosecretion, but also an increased depolarization. So the polarization of the cell increases because potassium is flows out of the neuron. And GIO, on the other side, the alpha subunit of the G protein, travels to inhibit adenylyl cyclase, the enzyme that converts ATP into cyclic AMP, so stops that particular intercellular signaling pathway. The same agonist can also activate the beta-arrestin pathway, which leads to activation of ERK1 and 2 protein kinases. Some agonists are biased in that they can favor one pathway, and other agonists are biased and they can favor the other pathway. But in the case of THC, there is very little bias. So I love this image. Was produced in 1990 by Miles Herkenam at the NIH. The receptor was cloned, that CB1 receptors were cloned that year. And this image was produced by using a radioactively labeled form of the cannabinoid ligand CP55940. And the reason why I love this image is because, well, first of all, is in a way for me, the face that launched 1,000 ships. When I saw this, I decided to start working on endocannabinoids. was certainly one of the major components, because look at how much cannabinoid receptors does a rat have. This is, of course, a coronal section, sorry, parasagittal section of the rat brain. You can see here the cortical mantle, the striatum and substantia nigra, the interpedunculair nucleus, and the cerebellum and the hippocampus. All these contain very large amounts of cannabinoid receptor. Indeed, the cannabinoid receptor is the single most abundant gibberellin coupled receptor in the mammalian brain, 4 times more abundant than dopamine D2 receptors, and 11 times more abundant in opioid receptors, mu-opioid receptors. So it's a very important, very active receptor. What does it do? It's pretty ubiquitous, in fact, but is mostly, not exclusively, but mostly found in presynaptic nerve endings. So this is actually a beautiful image generated by Istvan Katona, who at the time was in Hungary, now is in Indiana. And what you see here this is immunogold staining of one synaptic ending here. You can see it here. I think you can probably even see some of the vesicles. Each of these blobs is actually a receptor, and this is a GABAergic synaptic ending. In fact, we know from now myriad studies that CB1 receptors are localized not just presynaptically, but in a specific part of the presynaptic bouton, which is called the annulus or the perisynaptic region. So it's not at the active zone, is on the periphery of that. And it's ideally located to stop the activity of calcium channels, acting therefore, in a sense, as a modulating receptor. In fact, in most cases, activation of cannabinoid receptors leads to decrease in transmitter release. There are some exceptions, but in most cases, that's what it is. So this is what the receptor is thought to do in general terms in the central nervous system. But we shouldn't be fooled by the fact there is so much CB1 in the brain. It doesn't mean that this is a brain receptor. In fact, whenever I think about THC as a drug, I like to think of THC as a drug that impacts every single organ of the body, because there are cannabinoid CB1 receptors in every single organ of the body. And on top of that, there's CB2 receptors also in the periphery that can be important, particularly in the immune system. So in white blood cells, including monocytes and T lymphocytes, B lymphocytes, in white blood cells, CB1 and CB2 receptors regulate the immune response. Actually, it's a pretty strong effect, as I will allude to in the last part of my talk. In blood vessels, CB1 receptors regulate vascular resistance and blood pressure. And this is actually one of the first things an inexperienced person who uses cannabis experiences is the drop in blood pressure that causes an immediate sympathetic feedback response that causes 2 things. Heart rate goes up and the subject panics. So those are kind of interesting things, little physiology that is often forgotten by neuropharmacologists like us. So in intestinal smooth muscle, of all places, CB1 receptors regulate motility. In peripheral neurons, both CB1 and CB2 receptors regulate pain processing. So there are a host, and this is a partial list of the effects of the CB1 receptors have outside the brain. So why do we have these receptors? I gave you already the answer before. We have an endogenous cannabinoid system. That's why this receptor is there. And if one looks at the structure of THC, it's a very hydrophobic molecule. It's a lipid-like molecule. It's produced through this polypeptide pathway I showed you before. And so it's not that surprising, although it was at the time when it was discovered, it's not that surprising that the endogenous cannabinoids, the structures of which you can see here, are actually lipid-like, lipid molecules, a lipid-derived molecule. So the 2 of them, you will find more in the literature, but these are the 2 that actually, I believe, are now established. Anandamide, which is the ethanolamide. So this is ethanolamine, this is an amide bond with arachidonic acid, which is a fatty acid with 20 carbon atoms and 4 double bonds. Here, the double bonds are all in the Z configuration. 2-arachidonyl acid glycerol, which you see here, is similar in that it has the same arachidonic acid component. Instead of an amide bond, it has an ester bond, as the name indicates, and instead of ethanolamine, it has glycerol, 2-AG. So there are small pharmacological differences between the 2 of them. Anandamide is a partial agonist, 2-AG is a full agonist. anandamide and 2AG are almost equally active. But I think the most interesting part is the biogenesis and degradation of these compounds, which are completely different. In fact, as you can see in this, I'm not expecting you to remember this or to go through this in detail, but basically what you're looking at is that in both cases for 2AG shown here in this sort of flattened way and anandamide shown here, we have a precursor in the membrane. So there is a phospholipid precursor that has no activity. And when a stimulus arrives, enzymes are activated, which now cleave this precursor, generating bioactive endocannabinoids. In the case of anandamide, this is the precursor. In the case of 2AG, this is the precursor. And this precursor is actually a novel precursor. It didn't exist before we started to study this. And instead, phosphonadionyl bisphosphate, which you see here, is a well-known phospholipid involved in all sorts of different signaling pathways. So once you have an anandamide and 2AG, they activate CB1 and CB2 receptors, as I said, but every signal that is worth its salt has to be destroyed after it works. Otherwise, it's no longer a signal, right? And there are two enzyme systems that are involved in this. In the case of anandamide, an enzyme called fatty acid hammer hydrolase, or FA, which produces arachidonic acid and ethanolamine. In the case of 2AG, an enzyme called monosylglycerol lipase, or MGL, which produces arachidonic acid and glycerol. And there are now inhibitors for each of these enzymes. For example, in the case of FA, this is URB597 compound. In the case of MGL, this JZL 184 compound. Now, there are other pathways through which the endocannabinoids can be metabolized, and they're not deactivating pathways. They're actually activating pathways because they can be oxygenated, just like arachidonic acid is, by cyclooxygenase and lipoxygenases, and the products tend to be biologically active, not through cannabinoid mechanism, through other mechanisms. So what I think an important area of research now is understanding how this further metabolization of the, or activation rather, I made up the word metabolization, doesn't exist in the English dictionary. Anyway, the further activation of anandamide and 2AG can lead to other interesting signals. So what is the physiological role of the cannabinoid receptor at a synaptic level? I alluded to the role of the cannabinoid CB1 receptor in terminating or maybe modulating transmitter release. Let me put a little bit more flesh around that skeleton. So this is the model that is now widely accepted. People erroneously say that this is the endocannabinoid mediated retrograde signaling. I like to be more specific. This is not what happens with anandamide. This is what happens only with 2AG. Anandamide is different. But with 2AG, it is true, and 2AG acts as a retrograde signal. So what does that mean? Basically, anterograde signaling is the normal form of neurotransmission that we have with glutamate, dopamine, et cetera, et cetera. Basically, an action potential invades the synaptic bouton, calcium enters, vesicles are secreted, transmitter is secreted through vesicle fusion, and the transmitter now acts as receptors on the postsynaptic spine, in case it's an excitatory, or in case it's inhibitory, not. But in this case, it's an excitatory synapse, as you can see. So now, retrograde signaling starts when anterograde signaling is finished. So basically, in the case of glutamate, for example, glutamate will spill over to the sort of perisynaptic, again, perisynaptic annulus or ringlet that is in the perisynaptic spine, in the postsynaptic spine. So in this sort of perisynaptic region, there is an agglomerate of metabotropic glutamate receptors, which you see here. This is mGluR5. Two enzymes, phospholipase C and acylglycerol lipase alpha, which are responsible for the cleavage of the 2AG precursor, which is phosphonadionyl bisphosphate. So when glutamate arrives here, it spills over here, so a little bit aside from the central axis of transmission, mGluR5 or 1 are activated. These are type 1 and metabotropic receptors are activated. PLC is activated, DGL alpha is activated, and a massive amount of 2AG is produced locally. This structure is actually held together by Homer proteins, and so we call it a signalosome. And it was discovered in my lab by Professor Kwang Muk-Jung. So 2-AG released in large amounts quickly goes to the presynaptic bouton where it finds the membrane. Now, a membrane for dopamine or glutamate is a barrier, but for a lipid, it's not a barrier. For a lipid, it's a highway. So it gets, the 2-AG gets in the membrane. At the point, it can laterally diffuse to the binding site for the cannabinoid receptor, which is right here in the membrane, as I told you before. And this leads in 95% of the cases to an inhibition of transmitter release. In this case, an inhibition of glutamate release. Now, this signal must be terminated. And this, I think, is really an example of how nature can be extremely elegant in this. So there is an enzyme, MGL, the enzyme I mentioned before, monoacylglycerol lipase, is presynaptically located. And 50% of it is in the cytosol, and 50% of it, the numbers are exactly right, 50% of it is bound to the membrane. And the reasons why it moves back and forth. But when 2-AG has bound CB1 receptors, MGL is there and can cleave it into arachidonic acid. Just by mass action, 2-AG is removed from the receptor and its actions are terminated. So we have really this beautifully designed, and not designed, evolved mechanism to regulate through this feedback inhibition activity at glutamatergic synapses, but also is believed at GABAergic synapses, although in the case, I think the situation is a little bit different, but also at synapses that release dopamine, serotonin, and other transmitters. So I wanted just to mention, for those of you who are interested more in the plant than they are in the endogenous systems, that there are plant-derived compounds in cannabis that can interact with the cannabinoid system and endocannabinoid system, not just THC. For example, CBD it can inhibit Pha activity. Pha is the enzyme that destroys anandamide, deactivates anandamide, or it can also indirectly modulate CB1 receptor activity through a mechanism that is not fully characterized. Other things like tetrahydrocannabinoid can act as CB1 antagonists, etc. So let me move on now in the next couple of, at the end of my presentation, I have about half an hour, about the work that we've been doing on the lasting consequences of adolescent cannabis exposure. So this is an area that is funded by P50, so a center of excellence, and another funded center of excellence, which actually is about to undergo competitive renewal at the end of the year. And what I'll show is a very small part of the work that was done in the last 4 years as part of this center. The name of the center is ICAL, which stands for Impact of Cannabinoids Across the Lifespan, ICAL. So the first question we need to ask is why do we care about what happens to the adolescent brain if an adolescent smokes cannabis? Well, we know very well, I don't think we need to go into any stats to know that cannabis use starts in adolescence. Often starts in adolescence, and a lot of kids, and I'll show some numbers later on, do use cannabis. So that could be beyond any moralistic consideration, which I don't think belong in this in our world of science. There are very important biological reasons why that is problematic, and the main reason is because the adolescent brain, as the title of this slide indicates, is actually still fully developing, and some landmark events are shown here. But the bottom line is that adolescent brain development, which I'm by means an expert, in my understanding, is mostly centered around the reducing the number of synapses to a process called pruning. And pruning means that basically, as the name indicates, very nice plant metaphor, it removes the synaptic spines and reduces the number of synapses to those needed in adulthood. And we know now that microglial cells are critically involved in this process of pruning, and they're critically involved, therefore, in adolescent brain development. So microglia are, of course, macrophages. They're the macrophages of the brain. They don't enter the brain during life, but they do during later in life. They only enter during during early development, but they are, for all intents and purposes, very similar to macrophage. They prune synapses during postnatal development, but they also, as the individual matures, they also surveil the adult brain parenchyma for anything that could be happening. New insults of any sort. It could be insults coming from outside, so viruses, bacteria, as well as insults coming from inside, for example, danger signals that come from the damage produced to neural cells or other cells in the brain. Now, work by Nefistella and others has shown that microglia express all the components of the endocannabinoid system. And I'm giving you this because obviously we became interested in, because THC is an exogenous compound and microglia is so important in adolescent development, we became interested in understanding if THC would impact microglia. So this is a beautiful image from a review by Marco Prince, who has studied the microglia heterogeneity using all sorts of technologies. And what this shows in a nutshell is that we begin our life, and this is of course a mouse, as embryos, we begin our life with a very large heterogeneity of microglia in our brain. This heterogeneity tapers off with time, is still fairly high in the neonate animal, and in the juvenile, which includes adolescence, is reduced, but is still there. Eventually, in adulthood is limited, much more limited. And there are things that happen, that can sway this normal route. For example, there could be diseases which would produce now context-dependent subsets of microglia, as you can see here, or in aging, which is, as the Latin said, senectus ipsa morbus, disease is, old age is a disease in itself, also can cause the generation of new forms of new subsets of microglia. So we obviously we are interested in this area here, right? In juvenile. So we want to know if we give THC, if a human is exposed to cannabis or THC, what happens to the microglia, and what happens therefore to what the microglia does in the context of brain physiology. But before I delve into the data, I want to convince you of one thing, that we cannot study this. We have to be careful when we study this. If we want to be relevant to anything, we have to understand what does it mean to use THC as an adolescent? What does it mean to use THC, period? Because often pharmacologists like me make the mistake of thinking, okay, I want to see a response. I'm going to give it as much drug as I can so I can get a certain response. But that is, of course, it's circular reasoning. We need to try and mimic, I think, what people do and see if that produces a response and what kind of response it produces. So first of all, let me tell you, cannabis use among teenagers is pretty substantial, but is stable. And that's I think, a very good piece of news. You can see here that there are some changes, but around 1997, it kind of reached this plateau situation where about 35% of 12th graders, maybe about 30% of 10th graders, and about 10% of 8th graders use it. Oh, interestingly, this tip you see here was COVID. Clearly being at home doesn't favor the use of any drug, including cannabis. Now, this flattens, by the way, just on a side consideration, doesn't match the risk. The idea of risk, kids who think that is risky to use regularly are decreasing in numbers, but there is no increase here in the use. And that, again, is really good news. But let's try to be a little bit more granular and understand. So these young people you use, how do they use it? What I'd like to point out, this is work by Alan Badin and collaborators, where they studied very, very important paper studying the patterns of adolescent use. You can see here that approximately 20% of adolescent users, so not 20% of adolescents, only those who use, actually use it daily. And they use it mostly by smoking. You can see here this this hatched bar, a few vape, you can see here, and very few used edibles, which is this little bar here. So maybe 20 to 22% of adolescent users use daily. And that's very important because that's my target population. I want to study that population because I think that is the population at greater risk. If you use it 1 to 5 days in a month, I think stands to reason to think it's probably not as dangerous. Now, I don't want to get ahead of myself. It may still be dangerous. We need to do the studies, but for now, I'm focusing on this 20% here. So how do I do that? How do we do that? We set up a model where we inject 5 milligram per kilogram THC to mice, rats of both sexes, and in between PND 30 and PND postnatal day 30 and postnatal day 43. This is the adolescent period in rodents. And what we did is two things. We did pharmacokinetic analysis of this because we wanted to know if the concentration reached by THC in plasma and THC-OH in plasma were similar to those reached in humans. I mean, we don't have adolescent data, but we have adult data. And the answer to this is yes, because the peak concentration, the Cmax here that you can see, is about 1 nanomole per milliliter, which is approximately 300 and something nanograms, which is what people sees in, researchers see in human users. There is a big span there because smoking has a lot of variability, and this is an intraperitoneal administration. The other thing important is that the time to peak is pretty similar to what happens in humans within 15 to 30 minutes. And the relaxation is also not too different. So the tapering is not too different. So the pharmacokinetics of this dose seem to match the human pharmacokinetics. So what about the pharmacodynamics? We ran 3 assays in this particular experiment. We looked at body temperature, which is a very sensitive response, and activates CB1 receptors in the hypothalamus, presumably, but also locomotor activity here and food intake. So you can see that food intake and locomotor activity are not affected in adolescent mice by this dose. And there is a reason for that. Adolescent mice actually are protected against THC, but that's a separate issue. However, there is a response, there is a hypothermic response. So body temperature drops about 1 degree centigrade. The maximal possible response is called MPE. The maximum possible effect is 2.5 centigrade. So this is a modest response. So we are giving a dose that is modest, very modest, and we are giving it for once a day for adolescents. These mice look perfectly normal. And you can see that if we now wait until PND 70, which is here, where most of our experiments were done, in our measures were done, you can see that there is no more THC in the brain. LOQ stands for lowest limit of quantification. Nothing can be quantified here. And in wide adipose tissue, where the THC is accumulated, that's a very important point. In wide adipose tissue, the relaxation is much slower, but by PND 70, again, there is no more THC. So the data that I'm about to show you are data obtained in a situation where we're looking not at residual effects of THC, but at persistent effects of THC, okay? And the conclusion, as I said, this is a low intensity use, a daily low intensity use. So what kind of experiments I'm going to show you? We're going to dissociate brain tissues, PND 70, 49, and some cases 120. I won't show you 120 data today. So 49 and 70 mostly. We do flow cytometry to purify the microglia, or we do something called mass cytometry by time of flight. And then we do all sort of other molecular interrogations. So the first thing that we need to say using this principal component analysis graph is that adolescent exposure to THC causes massive changes in microglia gene transcription. You see that if you compare vehicle, would be vehicle here and vehicle here at PND 49 and PND 70, or these are already, the transcriptomes, the microglia transcriptomes are different. And that makes sense because the microglia, as I showed you before, changes over time. But THC has its own effects, as you can see, and there is a rough equivalence between the distance between these 2 points and the amount, the transcriptional differences, and you can see it here in these volcano plots, because these volcano plots show that A PND49, so when there is still some THC in the system, we have about 2,000 genes up. A PND70 is only about 1,200 genes up. And there are about 2,000 genes up at PND49 and about 1,100 genes down at PND70. So what are these genes? The first thing that I want to share with you is that are genes involved in endocannabinoid signaling. And here is a heat plot showing some of the enzymes here. I don't want to go into main details, but you can see by the red and blue, sorry, and green colors that, for example, this enzyme here, fatty acid hydroxylase or FA, is very highly elevated at PND49. So FA, let me remind you, is right here, is the enzyme that converts an anandamide into arachidonic acid. So it's really high and remains high. This looks a lot less, but it actually still is significantly high. Other enzymes, other components of the system, like the cannabinoid receptors, which are encoded by the CNR1 gene, are high, and then they go down. So let me show you some graphs to get a better sense of this. So you see here in APLD, the enzyme involved in the production of an anandamide is downregulated and then slowly tapers off back to normal levels. By PND70 PND70 is still downregulated, but returns to normal values of PND120. FA is the same thing. FA is very high at expression is very high, transcription is very high at PND49, is still significantly higher at PND70, and then is normal at PND120. Now, these changes don't only affect the anandamide pathway, they also affect the 2AG pathway, in particular MGL, the enzyme that cleaves 2AG, one of the two enzymes. MGL is probably the most, not certainly the most important one. And MGL, you can see, is downregulated and it slowly returns to normal. So there are all sort of different things. You know, it looks like, you know, anandamide degradation is increased, 2AG degradation is decreased. So we don't know yet what are the consequences of this. We don't know yet exactly what are the consequences of this, but if you look at other genes, the first thing that, you know, is striking is that the genes involved in microglia homeostasis, the genes that make microglia, such as EGR1, MFB, Siglec, and these are TGF receptor genes, as well TGF and TGF receptor genes. These are, and maybe the color is not very clear in this slide, but they are downregulated with a few exceptions. And also a protein that is often used to mark microglia, IBA1, is downregulated at PND49 and PND70, so in a persistent way. And microglia morphology is affected, and you can see, for example, in this case, that maximal branch length is increased by THC, and branch length is lengthy branches are considered a part of the so-called surveillance microglia. So it's a phenotype or morphological phenotype that is typical of surveillance microglia, and whereas the opposite is more activated microglia. So it looks like this microglia might be more surveillance than activated. And it gets, this becomes clearer as we now look at host defense response genes. So these are genes involved in the immune response that microglia, in the response to external danger signals that the microglia must mount. And you can see that there are massive changes, really substantial changes, fold changes. They go almost to 5-fold changes. This is prostaglandin synthase, interleukin B, interleukin 6, CD36, and various toll receptors that are significantly downregulated. And if one does a Go analysis on this, you know, and bioinformatics analysis, indeed, there are tens and tens of genes involved in the response to bacterial agents, to lipopolysaccharide, which is a bacterial endotoxin, positive regulation of the immune response, etc. So it looks like the host defense response genes are downregulated, which is, by the way, the opposite of what we expected. We thought that it would be upregulated because some other drugs of abuse cause such an upregulation, but that's not what happens with THC. So just to mark the point, adolescent exposure to THC alters microglia function until young adulthood, until at least PND70 in some cases, we don't know exactly between 70 and 120, but at 70, the effect is still there. The effect does disappear with further aging, but 120, PND 120 for a rat or for a mouse in this case is our middle age. So it is important to understand that throughout the young adulthood of these animals, the microglia is significantly affected. So, and how? Functionally, the first thing that happens is that there is a disabling of the microglia response to bacterial endotoxin. This is lipopolysaccharide produced by gram-negative bacteria. Normally, in animals that have been treated with vehicle during adolescence, these are all enzymes of the innate immune response, proteins and genes, of the innate immune response. These genes are all upregulated fairly massively. And in animals that have been treated with THC, the upregulation is blunted, substantially blunted. And likewise, you can see here, I think I can move this thing over here, maybe, yeah. You can see here that IBA1, which is massively upregulated by LPS, is also upregulated in THC-treated animals, but to a substantial lesser extent. By the way, also the baseline levels are different, although it doesn't appear from this particular experiment. So the conclusion of this experiment is microglia in mice treated with THC during adolescence is unable to mount a normal response to gram-negative bacteria, which is not really exactly good news, but it's something that we did not expect. You know, this was not something that was even suspected before this. So we wanted to understand it a little bit better. So what we did, we tried to ask how microglia heterogeneity changed in adolescent THC treated mice. And so for that, we used mass cytometry by time of flight, which, and I'm going to the details of the technical details of the method, which are a little bit complicated, but basically is a multiplex form of flow cytometry that uses mass spec instead of fluorescence detection. So you obtain data like this. Basically, this is a t-distributed stochastic neighbor embedding data reduction method. And basically what you have to focus is on the color, and each color here represents a phenotype or a subset of microglia. And so if you move here, you will see that animals treated with vehicle during adolescence have this population of microglia most abundant, and that's a surveillance microglia. Animals that then treated with vehicle but now received LPS, all of a sudden there is a shift, and the surveillance microglia decrease, and this other now subpopulation increases, and this is the activated microglia. You can also see that this population here appears, was much less represented here, now is much more abundantly represented, and these are monocytes. So these are actually monocytes that enter after LPS administration, enter the not so much the parenchyma, but certainly the structure of the brain. So they would be under the pia. These are BAMs, are the broad border associated macrophages, which are at the border between the vasculature and the parenchyma. Now, if you look at what THC does, so this is adolescence THC. So again, THC is not on board anymore. We give LPS in animals that had received THC during adolescence, and you have to look very carefully to see differences, which is good news. It means that basically it's not that THC is changing everything; it is changing something. But we need to look at it in a different way. So what we did, we used a SPADE analysis, and the SPADE analysis basically pulls out individual subpopulations. And these 5 subpopulations here, these subsets here, number 1 to 5, the intensity of the color and the circumference of this of the sphere here, of the circle, indicates the difference between treated with THC in adolescents and non-treated with THC. And I'm not going to go over too much details, but these are the 5 clusters. And the thing that I want to attract your attention to is that they have significantly reduced levels of interleukin-17A, which is a very important, of course, component of the innate immune response, of the major histocompatibility complex 2 also, and also CD68. So there are other changes, but like increase in FA, for example, or decrease in CB1, interesting changes, and we don't quite understand what they are. But this is what it looks like. The microglia then is heterogeneous, has these different clusters, and these clusters are induced by THC exposure in adolescence. adolescence. So I wanted to bring out another important function of microglia I did not mention before, and that's its role in social stress. This may seem strange, but some of you in this group, including Professor Carter, know very well about this stuff. So social stress, which is, yeah, I don't have to comment about it, is very, very important. It really depends on microglia. The activation of microglia is necessary for the response that mammals mount to social stressors. And in particular, if the stress is chronic, then there are changes that can have a very strong mental health impacts. For example, impact on depression and other mood disorders. So what does THC do to the microglia response to social stress? So to mimic social stress, animal experimenters have developed a model where this big guy here is a CD1 mouse, and this small guy here is a C57 Black 6 mouse. And this is the cage where this guy lives. He's a retired breeder, so he's kind of a cranky old guy. And you put in this little buddy there, and he doesn't like that. So he starts hitting on him until this guy gives up and says, "Okay, I'm out." And then, you know, the researchers have to take the animal out and put it in this other side of the cage, where the big guy cannot access the small guy, but the small guy can still smell him. So this is the model of repeated social defeat. This is a striking model. Look at what happens to the plasma concentrations of corticosterone in this poor guy. They go up 5, 6 times. And the same is true for the interleukin-6 and other things. Now, of course, you already noticed what happens if you do this experiment in animals that have been treated with THC. And I don't think in 30 years I've been doing this, I've seen anything like this. The effect is completely gone. It's completely gone. And you know, we repeated it quite a few times because it was really not really believable. But that's what it is. It's completely gone. And if you look behaviorally, that's also very similar. This is the elevated plasmase test that measures fear of aviary predators. Sorry, I'm going to have to drink a little. Anyway, aviary predators, and you can see that there is a very strong, in animals that are exposed to social defeat, there is a very strong decrease in the time the animal spends in the open arms, which are, of course, where the animal can be predated upon by birds, and they prefer, of course, the dark. And in animals that have been exposed to THC in adolescence, on the other hand, there is no change. There is no change. Again, the behavior is stable. They don't feel like it's risky to go on this side of the EPM. Social interaction is the same thing. The time in the avoidance zone in a social interaction chamber is increased after exposure to the RSD, the repeated social defeat stress, in vehicle animals, but not in THC-treated animals. And you know, the vice versa, the time in interaction zone is decreased in these animals that are vulnerable. There are always a few resilient animals that they don't care. But it looks like all THC-treated animals are resilient in that they don't exhibit any behavior. So I'm about to finish. Basically, what I showed you is that frequent low-intensity exposure to THC in adolescents disrupts endocannabinoid signaling in microglia. That's the first set of slides. Impairs microglia reactions to gram-negative bacteria. And actually, we have data showing that also viruses or viral mimics fail to elicit a full response in those mice. And finally, and most importantly, I believe, disables adult response to social stress. So this may seem like a good thing. You know, you don't respond to stress, you're better off, right? But that's not a good thing, because one set of experiments I love to do is to see whether these animals have an impaired response also to more modest forms of stress, the forms of stress that are needed to build our neuroimmune system. So my fear is that studies are needed now based on this work to understand whether the ability of teenagers who use cannabis as they mature into adults and as they develop into adults, this ability is impaired, and therefore their ability later on to respond to stressful stimuli is also impaired. So just to finish, I'd like to thank all of you, but also I'd like to remind you that sometimes framing the debate on cannabis has been hard for the wrong reason. And I think our job as scientists is to try to help bring some, some evidence to build opinions with. And with that, I'll, oops, I'm sorry. It looks like I have an Academy of Forensic Medicine here that doesn't belong. All right, sorry about that. Anyway, I'll take questions if you have any.
Well, thank you so much, Dr. Piamelli. That was an incredibly wide-ranging talk, and the new data that you showed at the end are really interesting. While we wait for Q&A, we do have a question, but there was a follow-up question that indicated that you had answered it. So I'm going to ask you a question, and we'll see. Here we get some more ones. All right, so before I ask you mine, Karen Bales, thanks you for the wonderful talk, and she's asking, do we know if THC has the same effects on microglia in adults?
We know that it doesn't. Actually, we ran that experiment. We did exactly the same experiment, the same length of time, the same dose in adult mice. And when we ran the RNA-seq analysis, it was really funny because we had, you remember 2,000 genes, and instead we had 20 genes and 20 genes in the adult. So really in the adult is essentially non-responsive. Those few genes are probably noise, they are even irrelevant. So, and thank you, that is a lovely question, important question, because it demonstrates that whatever we do in adults, the pharmacology of the adult is not the same as the pharmacology of the adolescent, just like the pharmacology of the female is not the same as the pharmacology of the male. And I think this lesson in biology is hard on us because as scientists, we don't like all this complexity altogether. And it's hard on funders because now they have to open their pockets more if they want to get data. But I think it's where science is going now.
So thank you for that answer. While we wait for another question, I'll ask you mine. So those results, particularly related to gene expression and microglial morphology, are interesting, as you pointed out. Perhaps not what we would predict, but then that's the data, and it's very valuable. Were you able to see any evidence of changes in synaptic spines, dendritic spines, synapses, pruning related activity in your adolescent mice? Because from a risk for schizophrenia point of view, that's a highly relevant sort of pathophysiological mechanism.
That is a very, very important question. We have not done that experiment, but that is exactly the type of experiment we would like to do. Just to give you a sense, what we plan on, we are actually already, of course, I'm writing the renewal right now because it's 800 pages piece of document. What we would like to do is, one of the several experiments I'd like to do is precisely that, understanding what region of the brain these things take place. The data I showed you are basically whole brain data, right? But we know that microglia has regional differences. And there are two regions in particular where we are likely to focus our attention, and that's the prefrontal cortex and the hippocampus. Both of them are important for a variety of things, including schizophrenia. And the idea is that, are those microglia changed differently from what happens, I don't know, in the cerebellum or whatever? And if so, is there a consequence on the neurons? And that can be articulated in different ways. For example, by using single cell RNA-seq to see if the neurons change along with the microglia, but also, just as you say, look at the spines, because that is clearly more directly relevant. To what extent we will be able to actually do that experiment, I don't know. But certainly is something that we would like to do. So this was, by the way, a 4-year center. It was not a 5-year center, and we had both a major flood in our building and COVID. So I hope that somewhere there will be, a light bulb will go in the head of the reviewers and say, okay, these poor guys did do whatever they could do to get some stuff done.
Yeah, no, that's a great answer. Thank you. This next question is from Dr. Tyler Lesh from Psychiatry. Fantastic talk. I'm curious if there are any situations in which the immunosuppressive nature of THC might have a more positive influence, such as inflammatory conditions.
Yeah, in fact, most people would argue that what we show is a good thing. Like, oh, okay, so you don't respond to bacteria, you don't respond to is better. My answer to that is if you don't respond to bacteria because you have a better response, fine. But if you don't respond to bacteria because your system is unable to respond, that's not fine. But the question is certainly important from an acute pharmacology response. So clearly THC is an immunosuppressing agent and could be leveraged for some degree of anti-inflammatory and immune suppression for therapeutic purposes. But in adolescence is a different animal, and I cannot overemphasize how important it is to understand how different the response of adolescent animals and humans can be to drugs such as that. So, with adolescents, I would argue that many of the drugs that are given, psychiatric drugs that are given to adolescents these days, should be revisited carefully, just because sometimes, one needs to see the negative effect, one needs to wait 10 years.
Yeah, so that's such an important point. So thank you, Danielle. We're at time. This was an excellent talk. I think you're probably going to get some follow-up questions from some of the participants here.
Happy to answer them.
I will just let the group know that our next webinar will be on May 12th at 11 a.m. It will be given by Houston Wilson, Assistant Cooperative Extension Specialist in Entomology at Riverside, and Katie Britt, who's a postdoctoral scholar there. And they'll be presenting on developing integrated pest management for cannabis in California. And I hope you all join us there. And I thank you for your participation. And once again, Dr. Piamelli, we'll see you all again soon.
Bye, everyone.